When you compare the genomes of any two individuals, you will typically find between 4 to 5 million differences, which are called genetic variants. The vast majority of these are polymorphisms and are shared with millions of other people. The most frequent form of polymorphisms are called single nucleotide polymorphisms, also known as SNPs. They are substitution variants, thus a variant where one nucleotide is changed by another one and must be found in at least 1% of the population. More than 650 million distinct SNPs have now been collected by the Single Nucleotide Polymorphism database. Each SNP can be distinguished by its own reference SNP identification number. If you look at the information for a particular number, you find that two alleles are mentioned: the reference allele, which is the nucleotide from the reference sequence, and the alternative allele, which is the genetic variant. Some SNPs have been associated to certain traits or disorders using genome-wide association studies, better known as GWAS. In these studies, researchers look at the association between SNPs and a common trait or disorder. When a SNP is found associated through statistical analyses, there is a relationship between the genotype and the trait or disorder. In other words, the alternative allele is statistically found more often in those that have the trait or disorder compared to those that do not have that. Remember that we are talking about associations and not causations. GWAS do not show causal relationships, like analyses of rare variants for Mendelian disorders do. Individuals may carry the alleles associated to a common disorder without ever getting that disorder. This is due to the complex nature of common traits and disorders, as we mentioned before. Depending on the trait or disorder, hundreds of SNPs can be associated to a disorder. The effect size of one SNP is thus relatively small, unlike rare pathogenic variants that cause Mendelian disorders. So why is the effect size small? SNPs are often located in intergenic regions and affect genes indirectly. For instance, a SNP associated with a disorder that is located upstream of a gene may change the expression of a gene by disturbing a sequence that is used for regulation of expression, such as an enhancer region. So while this SNP does not change the protein itself, as the coding sequence of the gene remains unaltered, it may influence the amount of protein that will be produced in the cell. SNPs rarely change the protein itself and are not disruptive for the function of the protein. If they would, it would be detrimental for many individuals, as more than 1% of the general population also carries it. A SNP may thus modify the functions of the DNA sequences where they are located. In essence, they make up the control panel that makes every individual unique by fine-tuning the number of genes that are expressed. SNPs are not disease-causing on themselves, but if one carries a combination of multiple disease-associated SNPs, one can have an increased genetic predisposition and therefore a higher risk of getting a particular disorder. So that person who gains weight very quickly may have a combination of SNPs that contribute to the propensity of gaining weight.